Phagocytosis is largely responsible for the depletion of cells coated with antibodies. Cells opsonized with the IgG antibodies are recognized by the phagocyte Fc receptors, which are specific for the Fc portions of some IgG subclasses. In addition, where IgM or IgG antibodies are deposited on the surfaces of the cells, they may activate and complement the system by classic pathway. Complement activation generates the byproducts mainly C3b and C4b, which are deposited on the surfaces of the cells and are recognized by the phagocytes that express receptors for these proteins. Antibodies that react with antigens present on the cell surfaces or in the extracellular matrix cause disease by destroying cells, triggering inflammation, and interfering with the normal functions. So the antibodies may be specific for normal cells or tissue antigens, or for exogenous antigens such as chemical or microbial proteins that bind to cellular surface or tissue matrix. The antibody-dependent mechanisms that cause tissue surgery and disease are illustrated and followed. So examples of antibody-mediated diseases for type 2 hypersensitivity are the following.
So to continue, during a hypersensitivity reaction, the body can also respond to its own components that are usually ignored. In a type 2 reaction, the response is caused by the presence of certain antibodies. Under normal conditions, antibodies are produced during an adaptive immune response to fight off pathogens. In type 2 hypersensitivity, the antigen is located on the cell surface or is part of the connective tissue. This leads to an antibody-mediated immune response to the body's own cells. The antibodies initiate a cascade of events that can lead to cell lysis, phagocytosis, or apoptosis. Therefore, a type 2 hypersensitivity reaction is also called an antibody-dependent cell-mediated cytotoxic response. Antibody binding to the antigen primarily activates the complement system. This system comprises specific plasma proteins that act as a cascade, mediating further activation of immune cells and heightening the intensity of the response. The complement cascade is triggered by a complement protein that binds to the antigen antibody complex on the cell. This leads to further activation of further complement proteins through cleavage of small fragments. These fragments are chemotactic, meaning they attract immune cells such as neutrophils, which binds to the antibody via Fc receptors present on the surface. These Fc receptors can detect and bind to the Fc portion of the antibody. After binding, the neutrophils generate aggressive substances such as radical oxygen species. So these substances cause cellular damage, inflammation, and eventually cell death. It is important to note here that hypersensitivity type 2 reaction is an antibody response against an antigen on the surface of cell membranes. These antigens can be, for example, first, a particular drug such as penicillin, hydrochlorothiazides, and quinines, which are rare cases and can be found on the surface of red blood cells and platelets. Second is an antigen could be a part of a bacteria. And lastly, another could be the rhesus antigen or the Rh antigen, where a rhesus negative person will mount an antibody response against a rhesus positive red blood cell.
Some screening tools used to screen for hypersensitivity. So patient history. Patient history is used to gather data about the patient so that we'll be able to make an appropriate care to the patient and know what caused the hypersensitivity of the patient. Screening tools for hypersensitivity, part 2. So we have CBC. CBC is the complete blood count. It is used to check whether or not your blood is within normal limits. So screening tools used to screen hypersensitivity, part 3. So we have the skin prick test. Skin prick test is used to identify what types of allergy you have or you are sensitive to so that the medical professionals will know.
What you been up to, Aiden? Yo, yeah, I've been dancing in my room, swaying my feet.